So today we're going to be talking about structure and function of DNA, talking about how DNA copies itself in the process of replication, and also get into talking about another form of nucleic acid called ribonucleic acid, or RNA. So first, let's talk about a nucleic acid. We haven't talked much about the fourth major macromolecule. We talked about the other three quite a bit with proteins, lipids, and carbohydrates. So nucleic acids are the fourth major macromolecule, and they are made of subunits or monomers, just like every other macromolecule. Like all organic compounds, they contain carbon, hydrogen, and oxygen, but nucleic acids also contain nitrogen and phosphorus. Now, the monomer that makes up a nucleic acid is called a nucleotide, and they're linked by bonds that are very, very strong, and these are covalent bonds. So this is a pretty strong and durable molecule. But as we see, we're going to talk about the structure of DNA. There's a couple of different bonds in DNA that you'll have to pay attention to because it'll be quite important for the process of replication. So a nucleotide, that's the building block of nucleic acid, is basically made up of three parts. Now, depending on the nucleic acid you have, the nucleotide might look a little different, but regardless of the nucleic acid, a nucleotide is made up of a phosphate group, a sugar, and a base. The phosphate group is pretty much going to be the same in all nucleotides. Doesn't matter if you're talking about a DNA nucleotide or an RNA nucleotide. The sugar will change depending on the type of nucleic acid you have, and the base might change. We'll get into more of this as we kind of continue here. So, we talked about the functions of the other macromolecules. The function of nucleic acid is for storing and transmitting genetic information, more technically called heredity. This is basically things like eye color, hair color, genetic diseases, etc. But more specifically, we're going to talk about how DNA and RNA codes for your traits. And specifically, it's the code. The code is the instructions for building every protein that your body will ever need. So, when you're looking at DNA and RNA, we're going to look at the differences here. But DNA stands for deoxyribonucleic, all one word, acid. And the first part of the name is actually named after the sugar called deoxyribose. So remember that a nucleotide is made up of a phosphate group bonded to a sugar. So the sugar in DNA is deoxyribose, which is why we get the deoxyribo part of the name. I will tell you that one of the big mistakes students make a lot of times is they call it deoxyribose nucleic acid. That's incorrect. So the "-ose" gets removed and replaced by that word nucleic. RNA is ribonucleic acid because it has a ribose sugar. So again, it's named after the sugar that's part of the nucleic acid. Okay, so we're going to focus on DNA for a little bit here. DNA is a double-stranded combination of nucleic acids. So we're going to talk about DNA kind of like an upright ladder. And we're going to take that upright ladder and we're going to twist it into a corkscrew, and that's going to be what DNA looks like. The two sides being one half of the ladder, the left side of the ladder, and the right side of the ladder. And then both halves of the ladder are connected by the steps or the rungs that go in between it. So that twisted ladder shape is known as a double helix. And the upright parts of the ladder are made of alternating deoxyribose sugars and phosphates. And the rungs, or the steps of the ladder, are where you're going to find the nitrogen bases. Now, in the 1950s, the actual structure of DNA was discovered by two scientists, James Watson and Francis Crick. They made a hypothesis that DNA was made of these two chains that wrapped around each other. Now, they used a lot of other scientists' work to come up with that hypothesis, mainly Rosalind Franklin and Maurice Wilkins. And some say that Watson and Crick, who are really given they're given credit for discovering the shape of DNA. Some say it really should be given, more credit should be given to Rosalind Franklin and Maurice Wilkins, who aren't really ever mentioned in the structure of DNA. And the reason is because they actually created the X-ray diffraction photographs of DNA, which was really the first pictures of what DNA kind of looked like. So this is just a picture of what DNA looks like. Again, you've got the edge of the ladder here twisting around. That's one edge, and then of course the other edge coming back this way. So that's the sides of the ladder. The rungs, or the steps, that's these here. These are made of the nitrogen bases. So you've got phosphates. Phosphates are in between the rungs. So this would be a phosphate because it's not connected to a rung. Then this would be a sugar, and then this would be a phosphate, and this would be a sugar. So you can see it alternates up and down the rung, or up and down the sides of the ladder. And then the rungs in here, we'll get into more of them here in a little bit. But notice there's one type of nitrogen base here connected to another nitrogen base here, and there's a bond right here that holds those rungs together in the middle. Now this is not going to be a very strong bond. This is not a covalent bond. The covalent bonds are like here, so they connect the nitrogen base to the deoxyribose, and they're here, they connect the deoxyribose to the phosphate. So this is a really strong bond that holds all this together. But this bond right here is not very strong, and that's going to be pretty important when we move forward and talk about replication. Just another picture of it. So kind of taking the double helix and you untwist it, and now you can see the ladder structure. Again, phosphate, deoxyribose, phosphate, deoxyribose. These are the rungs, okay? And again, on the other side, phosphate, deoxyribose, phosphate, deoxyribose. When you look at the actual molecules, you can actually see things a lot better. This is the phosphate group. So you've got phosphorus bonded with 4 oxygens. That's bonded to the deoxyribose. That's a covalent bond right here. This is deoxyribose. You can tell it's a sugar. Remember, it ends in ose. So it's going to be a sugar, and it's got that ring structure like all sugars do. Okay, so there's deoxyribose. There's another covalent bond that bonds it to a nitrogen base. This is thymine. Okay, the T stands for thymine. And notice that the thymine is held together to the adenine with a pretty weak bond, and we'll finally name that bond, called a hydrogen bond, which we did talk about way back in polarity with water. So it's a semi-strong bond, but not nearly as strong as like the covalent bonds. But again, so you got phosphate, deoxyribose, phosphate, deoxyribose. That's the upright part of the ladder. Over here, same thing, deoxyribose, phosphate, deoxyribose, phosphate. And then you've got the rungs of the ladder, the steps. Those are going to be the nitrogen bases. Okay, just a quick video just to kind of show you. It just kind of shows you what DNA looks like at the molecular level. Okay, so let's talk about the phosphates. Those are going to be the molecules that are in between the deoxyriboses and the upper on the sides of the ladder, on the upright parts of the ladder. Basically forms the backbone of DNA. It connects one nucleotide to the next. And the phosphates are very negatively charged, and we've learned hopefully that negatives don't like other negatives. So because they don't like each other, they basically repel from one another. And one phosphate doesn't want to be anywhere near another phosphate. They try to get away from each other, and they end up twisting the entire DNA molecule. So the reason that DNA has that double helix shape is because of the negative charge of the phosphates and the fact that they're trying to get away from each other, and by doing so, it just ends up twisting into a large corkscrew. All right, so now let's focus on the nitrogen bases. The nitrogen bases, again, are the rungs, and there's two groups. So you can take the nitrogen bases and separate them into two groups, purines and pyrimidines. Purines are double rings. So notice here is adenine. It's one of the purines, and notice it's made up of one ring there and another ring there. So it's a double ring nitrogen base. That's called a purine. And guanine, which is right here, guanine is also that double ring structure, 1 and 2. Okay, those are the purines. And then you have the pyrimidines. These are the single ring. So notice here's thymine, which is a pyrimidine, and cytosine, which is a pyrimidine. And notice that in DNA, a purine always bonds with a pyrimidine, and more specifically, adenine always bonds with thymine, and guanine always bonds with cytosine. And those are the rules. So when you're talking about code, and you're going to be decoding DNA as we go forward, remember that A's always goes with T's and G's always goes with C's. Notice also that it doesn't matter which side the A or the T is on. If A is on this side, well then T is on this side. If A is on this side, then T is on the other side. So because of those bonding requirements, we can always figure out how many bases are in a person's DNA if we just know one of them. So, and this is known as Chargaff's rule. Chargaff's rule says that if you know one of the bases, you can figure out the rest. So, imagine if you had 20% adenine. If you know somebody's DNA is 20% adenine, well then you also know that person's DNA is 20% thymine. Now together, that makes up 40% of the DNA. We know that obviously all of the DNA is 100%, which means that guanine and cytosine, in order to add to the 40%, has to be 60%. And because they have to be split evenly, we know then that guanine is 30 and cytosine is also 30. And that's known as Chargaff's rule. Another quick video here. Looks like this one I cannot play. Oh no, it does work. Okay.
DNA contains two strands of building blocks called nucleotides, arranged like a spiral staircase. Each nucleotide includes three parts: a phosphate group, a sugar molecule, and one of four bases: adenine, guanine, cytosine, or thymine. The sugar phosphate bonds form the double backbone of the molecule, the handrails of the staircase. But we find the genetic key to DNA in the steps of this stairway, the nitrogen-containing bases. These bases link up using hydrogen bonds in a very specific way. Adenine will bond only with thymine, A to T. Cytosine only bonds with guanine, C to G. While these basic pairings never change, the order of the pairs along each strand varies greatly from one species to the next. In this elegant design, we see how nature stores the instructions to build all living things.
All right, so now we're going to talk a little bit about how DNA copies itself. This is known as DNA replication. We talked about it in mitosis and meiosis, and that this happens during the S phase to make an extra copy of DNA so that when the cell splits, it has enough DNA to fill both cells. So our bodies go through what's called our cells go through what's called semi-conservative replication. This means that when DNA copies itself, you're going to see that the molecule is going to split in half, and then each half is used as a template to build another new half. So when you're all done, you're going to end up with 2 copies of DNA. And in the 2 copies of DNA, you're going to have half that DNA is going to be an old half, and half that DNA is going to be a new half. So replication occurs with a series of enzymes, a bunch of enzymes that are going to come in and unzip DNA and copy DNA in a very specific process. DNA helicase is the enzyme that's going to be doing the unzipping of the DNA. And it unzips the DNA right down the middle, right along those hydrogen bonds, because those are a lot weaker and easier to break. Then this creates what's called a replication fork. So the DNA doesn't all unzip. There's thousands and thousands of letters. So it's not going to all unzip and then copy all at the same time. So pieces of it unzip and then get copied and then come back together, and then another section is copied. And these sections are called replication forks. Now, DNA polymerase is going to come in. Once the DNA helicases come in and unzipped a section of the DNA polymerase is going to come in. And it's going to add bases. That would be the nitrogen bases, adenine, thymine, guanine, and cytosine, based on whatever half is there. So if it unzips the DNA and there's a thymine there, then it's going to bond a new adenine nucleotide, and then removes these RNA primers, which are basically placeholders. Now, replication happens, again, in segments. So at the end, the DNA segments are then spliced together by an enzyme known as DNA ligase. Again, it's going to do it in parts. It's going to copy certain segments, and then it's going to stick them all together at the end with DNA ligase. And again, this is a very specific series of events, and it happens during S phase, and it's going to copy all the parts of DNA in these segments and then splice them all together. This is just kind of a quick picture to kind of show how this semi-conservative replication happens. So you've got this color DNA here, and then it's going to unzip it down the middle, use each of that side as a template to make a new side. So then in the end, you're going to end up with half old and half new in both copies. Again, 2 copies of DNA.
DNA replication is defined as the process by which an organism's original DNA is used as a template for the production of a new complementary DNA strand. An enzyme called helicase unwinds the original DNA's double helix, creating a replication fork.
So you can see here come the nucleotides coming in.
Next, an enzyme named DNA polymerase III works down the leading strand and up the lagging strand of the replication fork, synthesizing 2 new strands of DNA by taking free nucleotides and pairing them with the complementary bases on the original DNA template. The process of DNA replication is described as semi-conservative because the 2 copies of DNA produced each contain 1 strand of the original DNA and 1 entirely new DNA strand.
All right, now we're going to switch gears a little bit and talk about ribonucleic acid. This is RNA. And this is really going to be important to understand for our next major discussion, which is protein synthesis or building proteins. RNA is a helper molecule. It's a nucleic acid. It's made of nucleotides. A slightly different nucleic acid than DNA. We'll talk about the difference here in a second. But its job is really to help DNA in the process of making proteins. And the difference is with RNA and DNA is RNA is only single-stranded. It looks like half a ladder. What this allows RNA to do is it makes it much smaller, makes it easier for the RNA to move around the cell. And obviously it's going to be a lot shorter because as you'll see when we start doing protein synthesis, only segments of the DNA are copied by the RNA. So it allows the RNA to then leave the nucleus but also move around the cell. Another difference is that as we kind of talked about earlier, because it's ribonucleic acid, the reason it's called ribonucleic acid is that it's made of a sugar ribose. So instead of deoxyribose being its sugar, it has ribose instead. There is one difference with its bases. RNA does not have thymine. Instead, it has a new base called uracil. Now, uracil uses the same bonding rules as thymine did, so uracil will bond with adenine. And adenine can bond with uracil. And like I mentioned, it's also much shorter in length. It's only about the length of one gene, and that'll be important when we talk about protein synthesis. Now, there are 3 types of RNA. All of them have slightly different jobs, but all of them, again, are helping DNA build proteins. There's messenger RNA, or short mRNA. This is a single-stranded RNA molecule that basically is going to copy down the information or the instructions from DNA. This happens in the nucleus. And then it basically moves out to a ribosome that's in the cytoplasm and brings those instructions out to the ribosome, because remember we talked about this in Unit 3. Ribosomes are where the proteins are built. DNA is located in the nucleus, so it's stuck there. It's also too large to leave the nucleus, and by leaving the nucleus, it could only build one protein at a time. This allows the DNA to stay in the nucleus, safe away from anything bad that could happen to it outside, but also allows it to make many proteins all at the same time by sending these strands of RNA out to many different ribosomes to build proteins constantly. Increases the efficiency of the cell. There's also ribosomal RNA. We don't really get into much on ribosomal RNA. It's rRNA. But this basically helps make up the ribosome with other proteins. So ribosomal RNA is kind of what a ribosome is. And then there's transfer RNA. Now, transfer RNA is kind of a unique RNA in that it has 3 very specific code letters, 3 very specific bases. And kind of a long strand, and we're going to talk more about this in protein synthesis, but basically it's got 3 specific letters that are really important, and then the rest of it is just kind of a long strand of tRNA that helps carry amino acids from the cytoplasm to the ribosome. So here's your 3 types. Here's mRNA. That's, again, single-stranded, long, single-stranded chain of bases. Ribosomal RNA is in here, and it kind of just binds, this is protein here, binds all this protein together, which makes up the ribosome itself. And then here's tRNA. Here's those 3 specific letters. Those are going to be really important. Those are called the anticodon, but we'll get to that in protein synthesis. And then the rest of the tRNA molecule is really just structure that holds on to this amino acid, which is going to be important for building proteins. If you remember, proteins are made of amino acids. And that's the end of our slideshow. Have a good weekend.